Hello, welcome to Bio 3. Uh, Today we will talk about DNA structure, DNA replication, and DNA expression. DNA expression has different names. Some people call it protein synthesis, some people call it transcription translation, some people call it central dogma. So let's start with the structure of DNA. Remember at the beginning of the semester, when we talk about the four major types of food, the four major biological molecules, nucleic acid is one of them. And one of the example in nucleic acid is DNA. We will talk about both DNA and RNA in depth in this lecture. But first, let's talk about the structure of DNA first. And we will start with a little bit of the history of discovering the structure of DNA. Well, I won't test you the history, but I always, when I cover this lecture, I will give you a brief history just to give credit to a female scientist. Officially, the DNA structure was discovered by James Watson, Francis Crick, and Maurice Wilkins in 1953. And these three scientists received Nobel Prize because of their work. Let's go back to these slides later. Okay, let's go over this now. DNA is a chain of nucleotides. We learned about this at the beginning of semester because DNA is a polymer. It is made of the monomer nucleotide. Okay. I hope you still remember the structure of nucleotide. Nucleotide consists of three subunits, three parts. Okay. This is important. A phosphate group, a pentose sugars. A pentose, that means it's a 5-carbon sugar. For DNA, we call it deoxyribose. We'll explain to you why we call it deoxyribose. From the name, we can tell that it doesn't have an oxygen. This sugar does not have an oxygen. We'll show you the structure later. And then, last but not least, we have the nitrogen-containing base. Or some types of call it nitrogenous base. It's the same meaning. So it's nitrogenous, nitrogen, and then NOUS, nitrogenous base. Nitrogenous base. Okay. So 3-part phosphate group, pentose sugar, and a nitrogenous base. So let's go back to the history of discovering DNA first. Yes, officially, Francis Crick, James Watson, and Maurice Wilkins are the people who first discovered the structure of DNA. That's the official story. But I want to give credit to a female scientist. Her name is Rosalind Franklin. At that time, there were many scientists competing to discover the structure of DNA, because they know that whoever discovered the structure of DNA first, those people would get the Nobel Prize. At that time, Francis Crick was like a principal investigator in the UK, and James Watson was the graduate student working in his lab, and Maurice Wilkins was the lab tech. And their neighbor is Rosalind Franklin. Rosalind Franklin is a female scientist. She had her own lab, which is very impressive. At that time, not many women do research or do science. She's one of the few female scientists. Not only that she has her own lab, that makes her very, very impressive. And her specialty is something called X-ray crystallography. What is X-ray crystallography? Basically, it's like taking X-ray film. Let's say if you go to have a chest X-ray, instead of taking your chest, you're taking the X-ray on your chest or on your lung. She took X-ray on different molecule. She take the X-ray in the crystallized structure of those molecules. That's why it's called X-ray crystallography. And she was the first person in the whole world who could take the X-ray of crystallized DNA. And guess what? The X-ray of DNA doesn't look like the X-ray of your chest. The X-ray of your chest, you can see your lung very clear. It looks almost identical to your lung, okay? But the X-ray of DNA looks very different. If you look at this is her DNA X-ray X-ray film for the DNA. So as you can see, it looks very, very different from the DNA structure that we now see all the time. It's called double helix, right? It doesn't look like a double helix. What happened is that at that time, only the very, very smart people, they can so-called decipher the structure of the molecule on the X-ray film. As you can see that the pattern on the X-ray film looked very different. And James Watson was one of the geniuses that who can decipher the structure of a molecule by looking at the X-ray film. And what happened is that they sent the lab tech to borrow the X-ray film from Rosalind Franklin, and then James Watson took a look at it, and then, according to his autobiography, he figured out the DNA structure by looking at it, but there's a little bit discrepancy in the math. You know, the distance between the atoms and stuff. So they couldn't figure out the discrepancy. And then one night, James Watson had a dream about the true structure of DNA. It turns out that the true structure of DNA is not a letter shape, it's a double helix. It is twisted. When the molecule is twisted in the form of double helix, then all the math, all the distance between the atoms, they fit perfectly. So, and after that they published the result. And then unfortunately, Rosalind Franklin, as you may or may not know this, in the past there's people have not discovered a way to protect themselves from radiation. You know, just like Mr. and Mrs. Curie, you know, the couple who discover radioactive isotopes. Those are the people, they died very early. Mr. Curie died very early, and then Mrs. Curie continued his research, and then she also died very early. But Mrs. Curie got the Nobel Prize. Unlike Mrs. Curie, Rosalind Franklin had never got the Nobel Prize because she also died early by working with the X-ray. So because she died early before people awarded the Nobel Prize, so the Nobel Prize was given to the lab tech Maurice Wilkin. Nowadays, it is almost impossible. No one would give any recognition to the lab tech. So that's why I want to give official recognition to this female scientist here. She deserved to get the Nobel Prize, but she couldn't get it. Yeah, she passed away too early. And then this is James Watson and Francis Crick. They look like this when they were young. Francis Crick passed away almost, I would say, more than 20 years ago. He moved to Caltech, and then he moved to California, and then he died here. And then James Watson moved to East Coast in the Long Island. He was the chancellor of a research institute called Cold Spring Harbor Laboratory, and then he retired to be the chancellor like in 2010 or something like that. 2010, 2009, I don't remember. So remember, this is James Watson, how he looked like when he was young. They were happy because they are getting Nobel Prize. This is how he looked like. Well, I had the privilege, to go to the Cold Spring Harbor Laboratory in, I forgot, 2010 or 2011, in the summer of 2010 or 2011, to take an advanced microscopy course. This advanced microscopy class taught me about the not just the compound light microscope, but also something called confocal microscope and also two-photon microscope. And at that time, James Watson already retired from being the chancellor in the Cold Spring Harbor Lab. And one afternoon, around lunchtime, I saw him eating on a table. So I asked him if he would, if I can have the honor to have a picture with him, and then he said yes. As a matter of fact, as long as you ask him politely, he will say yes all the time because he got used to it. For all these years, so many people ask him for pictures, so many people ask him for autograph. I also got a book and asked him to sign it, and he signed it. Yeah, he got used to it. So just if you have a chance to see him, ask him politely, and then he will say yes. So I just want to show off this picture. Well, your professor with a Nobel Prize winner. Yeah. Okay, let's go back to science. Sorry about all the sidetrack. Okay, DNA. As I told you that DNA is, I would say, the nucleotide, the monomer of DNA consists of 3 parts, right? One part is called the nitrogenous base, and we have 4 possible bases in DNA. You have to memorize this: adenine A, guanine G, thymine T, or cytosine C. Okay, A and G, they are called purine, and T and C, they are called pyrimidines. So DNA is a polynucleotide strand. That means a polymer. They form a strand. It's macromolecules with the backbone of alternate sugar and phosphate. Remember, the nucleotide consists of phosphate group, pentose group, and then a nitrogenous base. So the phosphate group and the pentose group, they form the backbone, and then the bases, the nitrogenous bases, what happened is that, so okay, you have one backbone here, you have one backbone, okay, and then so this is the sugar and the phosphate backbone, and then you have the base sticking out like this. You have the base sticking out like this. So these are the nitrogenous base. And then you have another sugar phosphate backbone here. And then you have also other bases, nitrogenous base here. So as you can see that when the two sugar phosphate backbone, they are parallel to each other, and then you have the nitrogenous base in between, forming a ladder structure. And then this ladder structure will twist. So they will twist like this. So it's like a double helix. So I hope you understand this part. My poor demonstration. Okay. So that is essentially the DNA. Don't worry, we have a picture to show you later. So well, I'll give you some time to think about, okay, which molecule that you have learned so far from this class has a more similar structure as nucleotide. Well, let's continue.